Thank you, Marty. Beck?
Are we all depressed? Wow. Well, as one of Gautam's slides revealed, we are in the midst of a perfect storm. On the one hand, we're learning about the power of the microbiome in keeping us healthy. On the other hand, we're learning about how we can use our conventional antibiotics to treat infectious disease and simultaneously, unfortunately, decimate that microbiome. On the third hand, if I had one, we have the resistome, which is feeding resistance solutions to those pathogens that we're so worried about. And on the fifth hand, or fourth hand, we have the pharmaceutical industry who has decided that chronic disease is a far more lucrative business model. And they're right, it is. So we should all be depressed. except some of us are microbial ecologists. And we continually remind ourselves that we live on planet microbe. We are nothing more than microbial fermentation chambers. So, rather than think about this challenge through the eyes of a physician or a patient, let's think about this challenge through the eyes of the microbes. And so let's look at this gorgeous artist hand-painted scanning electron micrograph, which Gautam also had on one of his slides. We I guess, have the same artistic senses here. And look at this. This is a human fecal smear. This artist hand-painted the diversity of the microbes. That's just a plant fiber that wasn't digested. Lovely, lovely, except for this little guy right here, a cyst of Giardia. Now, we particularly don't want that cyst of Giardia there, and so what do? We go to our doc, that's you guys, and we insist on an antibiotic, which you readily give us or oblige us with under duress. And we take that antibiotic and, oh my, not a pretty picture, huh? Remember, the challenge was to get rid of that guy. And as we all know, dealing with the microbial world and using broad-spectrum antibiotics means that we have the unfortunate, unintended, and unavoidable consequence of decimating the very microbiome that makes us so human. All right, now I'm very upset seeing that microbiome, so this particular patient is going to get a lot of probiotics, whether they work or not, and they're going to eat a lot of yogurt because now I'm more relaxed. Okay, we're back to the beginning. Now, let's ask the microbes how they would solve this challenge. What is the challenge? What is the challenge? Press the wrong button. It's that guy, right? That's what we want to get rid of. The rest of these are wonderful. They're our friends. Okay, so to do this, we're going to look at a genome-based phylogeny of bacteria. Okay, very similar to what David put up, but only one of the domains. This is just the bacterial world, and it's all based on comparing complete genomes. And I'm going to plot on there those lineages, and there are a lot of them here represented by genomes, of the bacteria that have successfully evolved and retained the use of broad-spectrum antibiotics, the precursors to what we have in the medical community, to mediate their interactions. And the answer is pretty quick. 2. 2. The Streptomyces, which were the boon to the antibiotic industry, and some bacillus. Now, so that guy shouldn't have shown up yet. In retrospect, of course this makes sense, because why would bugs produce antibiotics that kill the community they live in, right? They don't live in sterile environments like we force them to do in the lab. They live in communities. And killing your community is like burning down your forest or chopping down your tree. It's not a good idea, and therefore only some particularly strange microbes do it. So what are the other options, though? Let's ask the microbes. Well, in fact, there are a number of them. They're the bacteriophage. Are they live? Are they deployed by microbes? We don't know necessarily. Do we care? They can kill other bacteria. What about pheromones? Quorum-sensing molecules? What about my own favorite, which are the bacteriocins? And I particularly like bacteriocins, even though they're proteins. Oh my goodness, proteins as drugs, imagine that. Because if I ask the microbes, how do you choose to interact in your community What is the answer? Well, give me a minute. Brendan has to give me an extra minute because this takes so long. They all do it. The only things that's missing there are the ones we haven't looked at yet. And give me one undergraduate, a few $100, and I'll find that bacteria center, okay? And I will take donations. All right, but they're proteins. Goodness gracious, they're proteins. How could these possibly work? Well, we'll get to that in a minute, but first let's recognize the microbes are speaking to us. They're not using broad-spectrum antibiotics in mediating their interactions. They're using bacteriophage, pheromones, other small molecules, and there's these whomping big bacteriocin proteins. So, could we not begin to envision a world where we, like the bacteriocins and the phage and the pheromones, target our interactions? And I know the first answer. I got an email from David. We're all worried because we're symptom-based diagnostics, right? We don't actually know who the offending agent is when we walk into the ER or into the clinic. It could be a single bacterium. It could be many. What are we going to do? We have to save the patient. I give you that. Until we alter from symptom to molecular-based diagnostics at the bedside, we must retain these valuable tools to fight infectious agents in life-threatening situations. However, let's step back for a minute. I believe it was Marty or Don. Well, we all say the same things. I think we're starting to be a symbiotic organism here. 80%. It's hard for me to sit down. 80% of the antibiotic use has nothing to do with saving life-threatening illnesses, right? 80% of it. So let's start there. Let's see if we can't develop tools to deal with the agricultural-based, the veterinary-based application of pharmaceuticals. In addition, the human-mediated cases, most of them are for urinary tract infections, right? You guys write the prescriptions, I don't. You write them for UTIs, or for COWD, or for acne, or for ear infections or conjunctivitis. Less life-threatening. Often we know the 1 to 5 infectious agents involved. So let me end by illustrating, can we actually develop things like proteins as drugs? And the answer is yes. One, we are easy to find. If you were paying attention on the last slide, you give me the pathogen, and within 3 days I will find you the drug. And ImmuCell just challenged me that, and I won. And they're now licensing that drug to treat E. coli-based mastitis in cows. They are highly stable. I call them undergraduate proof. They make it through an autoclave. They sit on a bench for 6 months and don't degrade. I can add detergents. I can add acids, bases. These are not your average proteins, ladies and gentlemen. And most important, they're non-toxic to human cells. In fact, I'm on my way to Beijing in a few hours to see the results of a 2-year study of intravenous and oral application of these molecules, these 70 kilodalton molecules, through turtles, chickens, ducks, cows, macaques, and humans. The Chinese, as we noted earlier, are far ahead of us in this study. And I am going to finish on time. They're very low MICs, 200-fold fewer moles of molecules required to treat a disease than, say, ampicillin or penicillin, and that's because they have single-hit kinetics, and they're reusable. They kill a cell and they keep going. Better yet, they kill non-growing cells, so they kill in biofilms. Try to do that with an antibiotic. It's pretty depressing. Now, they have a range of specificities, everything from a single strain of E. coli 0157H7, which we have a solution to kill, versus all the gram-positives. And we can manipulate that specificity easily genetically because these proteins are just so cool. And then finally, if we change the paradigm from broad spectrum to targeted, we don't have the same selective pressures for resistance. And then, of course, finally, if they didn't work, I wouldn't be here. And that's why we have a small biopharmaceutical company trying. We don't want a biopharmaceutical company. I'm an academic. I want to keep doing research. But until I can prove to the pharma companies that these work, I have to keep going. So let me end by hopefully remembering a quote from one of my favorite authors. And the quote is, "Infection or disease is often the result of a miscommunication about the borders, a misunderstanding and a symbiosis, a failure of one side or the other to observe the boundaries." That's the situation we have been in. And I urge you to rethink, just as David Relman suggested in his introduction, that we must now understand that microbes are our friends. This is Planet Microbe. And if we hope to achieve parity with them, we've got to communicate in their languages. Thank you.